Hi, welcome to my presentation. I'm Willow Livingood, and I'm presenting the cell tracking challenge using the KIT GE inspired method on HeLa cells. So let's start with the background and the data set. The cell tracking challenge is a competition created to inspire new methods of tracking cell proliferation and migration over time to distinguish between healthy cells and potentially diseased cells. And the purpose of this is to help with early detection of certain diseases such as cancer. The cell tracking challenge was created by Carlos Ortiz de Soloranzo at the University of Navarra. And the method that inspired me the most was the KIT GE method, which was entered by Johannes Stegermeier and Ralf Miku with the Karlsruhe Institute of Technology in Germany. The dataset we'll be looking at is the for N2DL HeLa, which are HeLa cells stably expressing H2B GFP, which is used for imaging, and these were submitted by the Monotrike Consortium. This is a 2D dataset, and you can see some example frames below. On the far left, you have a frame from the start of the dataset at time 0. In the middle, you have a frame from the middle at time 46. And on the far right, you have a frame from the end at time 92. So let's move on to segmentation. In the upper left-hand side, you have the original image for reference. This was taken at time 0, and then pre-processing was applied. I used median and Gaussian filtering to help separate the foreground from the background. After that, I used Laplacian of Gaussian filtering, or LOG, and this is where my method and the KIT GE method differ. They use the LOG filtering to help find the center points of each cell. I use the LOG filtering to help find the edges of each cell. And I just found that when examining each of these methods by eye on this specific data set, the edge detection seemed to track more cells. And then once all the edges for the cells were detected, you had to fill in the cells. I used dilation to help fill in the edge borders, or erosion to help remove any edges that were detected that were too small to conceivably be cells. So in the video, you can see the segmentation on the full data set. And then moving on to tracking, KIT GE, their method used nearest neighbors. They created a custom MATLAB toolbox because this is a greedy method and they were working with large data sets. I recognized that, so I scaled their method down a bit. So instead of looking at multiple nearest neighbors, I just looked at the nearest neighbor. So for each frame of the image, the center points of the cells were detected. And then the center points of the cells were measured between the frame and then the next nearest frame. And the smallest distance between the two frames was the ID of the cell. So on the video on the left, you can see the identified and labeled cells, but because there's so many cells near the end, the labels get kind of chaotic and difficult to read. So in this next video, you can see that I use the ID to uniquely color each of the cells instead of labeling the image. So for validation, the cell tracking challenge comes with 2 benchmarks. They use the cell tracking benchmark and the cell segmentation benchmark. The overall performance of the KIT GE method for tracking was 0.870, and this was the average of the segmentation and average training accuracies. And for segmentation, they used a Jacquard similarity index with a 0.770, and for tracking, they used the weighted distance between the evaluated and the measured for an accuracy of 0.970. For the cell segmentation benchmarks, the KIT GE had an overall performance of 0.871 with segmentation of 0.770 and detection of 0.972. And so my plan for validation would be to enter the results of my tracking challenge into their evaluation software. And this would require editing my software to be able to produce their expected inputs. And then once results were returned from their evaluation software, I would statistically compare results against KIT GE using a 1-sample sign test, and this with a null hypothesis of no difference between my method and the KIT GE method. And this was selected because we can't assume any distribution of our accuracy scores entered into this sign test, and it's good for small sets of data, specifically where you have 6 scores for each set of KIT GE or my evaluation. And this concludes my presentation. Please let me know if you have any questions.